The views expressed on Skeptically Speaking do not necessarily reflect the opinions of the University of Alberta, the Students' Union, or CJSR.
Hello. Welcome to Skeptically Speaking, the show that asks you to call in and question everything. I'm your host, Desiree Schell. I've been doing some thinking this week. When we first started doing Skeptically Speaking, we knew that we didn't know everything. That's a given. But throughout the 37 episodes that we've done so far, this is number 38, our understanding of just how much there is that we don't know has only increased. And this is a good thing. Because realizing that you don't know something gives you the chance to find out. No one looks for answers if they think they already have them all. And I don't know if you've noticed, but there seems to be a serious lack of people who want to admit that they don't really know much about a subject, and they probably shouldn't have an opinion at all. So I will remind us all right now. It is not mandatory to have an opinion on everything. It's okay not to know. None of us can be experts in every subject. All we can do is try to find out what the people who are experts are saying, what the body of their research has shown, and where the evidence lies. It's not 100% foolproof, but it's the best that we've got. And that is why we here at Skeptically Speaking have so many questions. And that's a long, roundabout way of introducing today's topic, microbiology. Lisa Hammett will be on the line to explain a few of those things that we don't know about the things that we can't see. But first, to speaking up. We have Mike Meraz with us today from Ventura, California. Professionally, he directs university enrollment, outreach, and marketing programs, as well as support programs for young scientists seeking graduate degrees and leadership positions in scientific fields. With degrees in geophysics, education, and counseling, he is particularly interested in efforts which bridge the gap between science and the general public. He's currently taking a break from the academic world to raise his son and using the very little extra time he has to contemplate contributions to the skeptical community. He's recently released the first episode of a new podcast, Actually Speaking. Welcome to Skeptically Speaking, Mike.
Hi, thank you.
So tell us about the podcast.
Well, it's actually something very different from what people are used to on skeptical podcasts. Taking a risk and setting the science aside and exploring the human side of skepticism. I like to sum it up best by my little tagline, how to live skeptically and still have friends, because I think some of us actually struggle with that, and I know I do. But it focuses on interpersonal communication skills needed to actually promote critical thinking. And an important point to know about it is that it's really not about science. It's about our interactions with non-skeptics. And this actually comes from my own interactions, my own frustrations with family and friends. I'm only recently a self-proclaimed skeptic. I wish I had come to this conclusion earlier in life, but it came a little bit later. And I had many years of arguing and fighting and strained relationships with my family. One of my favorites is alternative medicine, and psychics play a large role in my extended family. And so holiday gatherings and different family gatherings tend to be kind of fun and a little stressful. And actually, over the years, I've damaged a lot of relationships and really watched communication stop between a lot of family and friends. And when that happens, there's really not a whole lot of critical thinking happening anymore. So I really wanted a way to be able to live skeptically, but in a healthy way. And I wanted to share my skepticism without shutting people down in my daily life. I think we all want this. And lots of people, tell us what we should be doing as skeptics, how we should be portraying a more positive image. And, taking more care in how we interact with people who don't quite see the world in the same way that we do. But there aren't very many resources out there telling us how to do that. And I really wanted to know more about the human side of skepticism. So I decided to do it myself. And I created the Actually Speaking podcast. And in the podcast, I started off very simply with just having two goals. I wanted to, one, know how to live skeptically while having positive, healthy relationships. And I wanted to be able to share skepticism in a way that promoted critical thinking. And so I designed the podcast around that. Everything revolves around those two goals, and it's intended for daily life, family and friends. This isn't really geared towards someone who's in the media who has more of a leadership role. It's just, for the average skeptic in their daily life. And so I got two episodes out now, and the one that just came out was about realistic expectations. And some of the other ones that are coming up are civility, which is actually a really favorite topic of mine because I think there's a lot of confusion around it. Just practical skepticism, thinking critically, social networking, childhood fantasies, the whole tooth fairy, Santa Claus, and things like that, that we kind of struggle with because it kind of conflicts with our skeptical lifestyles. And one of the things that's been requested a lot from people who have written in that started following the show a little bit early is about parenting. So I have a 6-year-old, so I'm still relatively new to parenting, so I'll need to brush up on that a little bit.
And that can be a landmine right there.
It can, and it also sucks up a lot of time. But the podcast really, it's focusing on the skills and the application of those skills. It's a very short conversational approach to communication skill training. It's every other week, and it's my own learning process. That was something that was real important to me. I didn't want it to be just a lecture. I wanted it to be me figuring the stuff out for myself, and through the podcast sharing it along the way, because I want skeptics to be able to share that factual and scientific information that they have more effectively. Because I think how we share it will either open or close lines of communication. And that's important. That really matters. So I'm hoping that the podcast will complement the other science and skepticism podcasts out there and help skeptics live openly skeptical lives that are compatible with their family and friends and relationships and promote critical thinking.
So do you think that effective communication is a skill that's especially necessary in our community right now?
I think so. One of the reasons it's necessary is because, just talking about the skeptical community, it's changing. The skeptical community is a lot more enabled and active. The internet is really changing the whole dynamic of how we share information, how we interact with each other. And as a skeptical community, we want to be able to expand. And if we don't have the appropriate skills from doing that, we're actually just shutting people down instead of promoting critical thinking. Mm-hmm. And within the skeptical community, we hear over and over again that skepticism needs to be more appealing to the mainstream.
Right.
We need to break those boundaries and expand. I know you went to TAM, and we heard a lot of that this past summer. And I completely agree with that. But so my own take on that, or at least the avenues of doing that I'm trying to explore personally and share, is that one of the things is that I think we need to pull back a little bit and go back to our roots of critical thinking. I think sometimes we focus a little too much on science, and I know some people will disagree, but critical thinking isn't specific to one particular discipline. It's in journalism and politics and law. There's a lot of ways people can think critically. It's not always just in scientific fields. And I think opening that up a little bit would make the community more approachable. And I also think that skepticism needs to be more practical and applicable to people in their daily life. Some people call that dumbing it down. I don't see that. I really don't see that. And teachers have been dealing with this for a long time. I mean, any teacher that's been in the classroom and sees the glazed look on the kids' eyes, when they're teaching chemistry or physics or math, they learned a long time ago that the way you get past that is by making it applicable and practical to daily lives. And the last thing is that skepticism needs to be approachable. It needs to be less threatening, more positive, and it really has to be. We have to cultivate a good image for the skeptical community.
Well, are you opposed to the more bombastic and confrontational skeptics among us?
No, not at all.
Okay.
Not at all. And actually, when I mentioned civility, it's actually one of my favorite topics because civility keeps getting thrown around a lot in terms of whether or not we should be civil or we shouldn't. And no, we need to be more forceful. No, we need to be kind of nice. But I think there's a fundamental misunderstanding about civility. It's often confused with being passive or non-confrontational. And actually, that's not true. In order to be civil, you need confrontation. It's kind of like you can't be courageous or brave unless you're fearful. Mm-hmm. One needs the other. I mean, civility, just by definition, is just a matter of being polite. It's really just a simple courtesy. So being polite and respectful in your exchanges and your criticisms and your confrontations with people applies whether or not you're assuming a supportive role or a more assertive role, more of an activist, because we really need both. And actually something that we had talked about before that's applicable to this is a theory of personal growth called revolving around challenge and support. Now it really ties in with that aggressive and supportive side of the skeptical community, because those that work with people to foster personal growth know that people grow most effectively when they're both challenged and they're both supported.
Right.
Too much challenge and you fail. Too much support and you never move anywhere. You never change. And for people to grow, you actually need a balanced combination of both, of challenge and support. Well, you can take that out of the individual level and you apply it to a non-skeptical community. For that non-skeptical community to grow, they also need to be challenged and confronted on their certain beliefs, but they also need to have a support mechanism in place so that they can safely explore, those skeptical waters. And that's where the two different roles, the more aggressive role and the more supportive role in the skeptical community, really need to work together to balance each other out. And they're both valuable, and both of them can be civil in the process.
So now I'm just curious, what would your ideal skeptical community look like then? Oh,
that's a tough one. The ideal skeptical community would probably a community that's segmented, that has the skeptical lead, that has, well, I don't want to put labels on it, but has a component that's dealing with the media, has a component that's dealing with, that has more of a leadership roles, that's challenging more widespread beliefs, with the broader strokes of what needs to be done to confront misinformation, harmful information, and to challenge it and to just eradicate it where possible. And the opposite end of the spectrum would be a group of people that are doing similar work, but on a more personal level, within our daily lives, going through and making sure that people have an opportunity to grow and think critically, so that all the misinformation that's out there, if you think of it kind of like a disease, like a virus, it slowly loses any footing. It has no place to spread. Its food source, its livelihood is being taken away. And if we attack the issue on both ends, the two complement each other. And I think a community like that would be ideal. But of course, the reality is that there's a lot of other complications that come into it, and we all get kind of messy and make mistakes. And, the skepticism is about learning. It's a human process of learning. So, like I said recently, there's no finish line to a skeptical lifestyle. You're never done. Whether you're just beginning or you're wrapping up a career of skeptical living, there's always room for more learning, and there's always the possibility for mistakes.
I'm very excited to see where you're going to take this. I love the first episodes. So, where can people find this?
Well, they can find this on iTunes. Just look for Actually Speaking, or they can go to actuallyspeaking.com and get some more information there. And if they want to follow me personally, I'm on Twitter at Factually. That's with an F.
Perfect. Thanks for being here, Mike.
Thank you, Desiree.
And if anyone in the audience has a new skeptical project on the go, or an event to promote, or a topic that deserves an audience, this is your chance. Send us an email with a brief outline of what you'd like to talk about to feedback@skepticallyspeaking.com. Put speaking up in the subject line, and we'll see what we can do. And we'll be right back with Lisa Hammett on microbiology right after this.
You are listening to Skeptically Speaking, thanks to the wonder of podcast technology or the generosity of one of our rebroadcasting stations. Skeptically Speaking is broadcast live out of CJSR 88.5 Edmonton, Alberta at 6 p.m. Mountain Time every Friday. If you're not in Edmonton, you can still listen to the broadcast live in our Ustream.tv feed, which you can find by following the Listen Live link on our website at skepticallyspeaking.com. You can also participate in the chat room and submit questions. During the week leading up to each show, you can find information on the upcoming episode and guest, and submit a question to be asked on the air. You can also subscribe to the podcast on iTunes. Our deepest thanks go to the stations that donate their valuable airtime to our program, including LP 99.1 FM in Madison, Wisconsin, Public Reality Radio WPRR 1680 AM in Grand Rapids, Michigan, Roslyn 101.1 in Roslyn, British Columbia, CJAI 92.1 Amherst Island Radio Broadcasting Inc., and on freethoughtradio.com. And now, back to the show.
Welcome back to Skeptically Speaking. I'm your host, Desiree Schell. We're here today with Lisa Hammett. Lisa has a master's in microbiology from Auburn University in Alabama. She's worked for a biotech company, a forensic lab, at the National Institutes of Health, and in a cancer biology lab, and has taught biology and microbiology at various community colleges. Currently, she's a crazy minivan driving homeschool mama too. And she's a card-carrying member of the American Association for the Advancement of Science. She loves science fiction, Star Trek Next Gen, and yes, she was in band in high school and college. Welcome to Skeptically Speaking, Lisa.
Hi, how are you?
I'm well. Thanks for being here.
No problem.
So before we get to microbiology, I have to ask about homeschooling.
Okay.
So it's usually linked in my mind with religion, but that is not the case here.
Yeah, and that's the case here in what has been referred to as the buckle of the Bible Belt here in Georgia.
So why did you start homeschooling?
Well, I wasn't real comfortable with the public schools, and honestly, I don't think they focus enough on science and math because I think that science is not just a subject, but a way of thinking. And I think schools are too focused on trying to meet standards and memorizing facts instead of learning how to learn. So that was the main reason, I think, why I chose to do it. Never, ever in a million years did I think that I would. But here we are.
So what are some of the benefits and challenges you're seeing?
The benefits are that we can move along. We don't have to wait for, say, other students who are not maybe catching on as quickly. I can move along if they have the concepts, and there's no reason to beat the dead horse. The other challenge is that sometimes you just feel totally like you're out of your depth, that you're not doing it right, that when they turn 20, they're going to end up in therapy, and it's all going to be mom's fault.
Right.
And are they going to get into college? That kind of thing. And sometimes there's a little bit of judgment. People are saying, well, aren't they going to be socially handicapped and things like that? So you worry about those things. But overall, I think it's been a very positive experience for us.
Well, now I feel like I want to do an episode on public schools versus homeschooling because I know that there's a lot of people out there that would disagree with you on this topic. So I'm going to leave
absolutely.
That there, and we'll move on to microbiology.
It's definitely a personal choice.
Oh no, definitely. I'm very interested to hear what people's reasons are and the experiences they're having with it. But we will move on to the topic of the episode, which is microbiology. Okay. So in the 21st century, we here are used to the idea that there are living things too small to see.
Right.
But when did this idea come about?
Well, actually, it's been around probably for longer than you might think. I mean, not the idea that maybe living things were causing disease, but the disease could be transmitted, and there were sources of these diseases. For instance, in biblical times, you read in the Bible about leprosy and how they would quarantine people and shun them and force them out of their villages because they didn't know that these diseases could be transmitted. In Greek and Roman times, they also had this idea that little animals, they called them, could come in through the nose and the mouth and maybe cause disease, but they really couldn't pinpoint it. Then in about Robert Hooke, in about the middle of the 1600s, was developed a compound microscope, a very simple one, and he looked at cork, which is a wood, and he saw these little cells, and it just kind of went on from there. And most people are probably familiar with Van Leeuwenhoek, Anton van Leeuwenhoek, who is the Dutch man that really developed the microscope and put developed the lenses and was able to see at about 300x or 300 times what you can see.
Right.
And he saw these little creatures in water. He even looked at his own feces, his own mouth scrapings, and sources from sick people.
Now that is a scientist.
Yes. Truly.
So when about did they start realizing that microorganisms could cause disease?
Probably in the middle of Pasteur, I think he was the one that really started to show this germ theory of disease, because he came up with pasteurization, and the fact that he figured out that these microbes, yeasts and in particular, were fermenting juice to make wine. And it sort of set in this motion this idea that these microorganisms interact with organic material. And so then it kind of went from there to maybe these microbes are causing disease and that kind of thing. And so Pasteur and a guy named Koch kind of came up with this idea of these organisms actually causing the disease.
Okay, so we know when people first started studying them. So now, what does microbiology as a field cover?
Oh, lots and lots of stuff. It can cover virology, the viruses, mycology, the fungi, plants, a little bit of everything. Immunology, because diseases are caused by these viruses and bacteria, so it kind of overlaps with immunology. Molecular biology, because there's all this molecular aspect, the DNA and the biochemistry of it all. So biochemistry is in there when you're looking at metabolism. It's a little bit of everything. When I teach a class in microbiology, we have to cover chemistry, biochemistry, organic chemistry, virology, immunology, epidemiology, just a lot of ology, really.
Okay, well, let's start with 101.
Okay.
How is a virus different than a bacteria? Because I've been talking to a lot of people about this over the past week, and I realize how a lot of people actually don't know this.
Right. Well, bacteria are much, much larger than viruses. In fact, bacteria have viruses that will infect them as well. Viruses are not really considered a living organism like a bacteria. Viruses are completely dependent on whatever their host is. They're very host specific. So like you're not going to get parvo from your puppy dog because they will only infect that species and usually only a specific kind of cell in that particular species. And they only really have a nucleic acid, either DNA or RNA. They don't have a nucleus, or they don't have all the other stuff that's inside eukaryotic cells, which are what our cells are, or bacterial cells. You can't kill them with antibiotics. Usually, for the most part, bleach will kill them, like household bleach will kill them. With bacteria, you can control those with antibiotics, alcohol, and some of the other antimicrobial agents. So in a nutshell, that's it.
Okay, and how about a fungi?
A fungi are generally eukaryotic. Well, they are eukaryotic cells, multicellular, and they're like yeast. Well, yeast is unicellular, but yeast is a fungi, and we're all familiar with yeast, and it makes bread rise, and it's the stuff that makes ethanol for your cocktails. And the other ones are the molds that grow on your bread when you've left it out too long, or what makes your lettuce turn into mush.
Is mush a technical term?
Yes, it is actually.
Sweet.
It is
I use
now.
Technical terms. So one of the things that I realized a couple major illnesses ago, before H1N1 and before the avian flu, we had mad cow disease. And
right.
I had assumed that was caused by bacteria, but in my homework for this show, I found out that apparently it's caused by a prion.
A prion or prion, yeah. And what that is an infectious protein. And as far as mad cow, Jakob Creutzfeldt is another variation on that. What it does, it can get in your brain, and that is the abnormal version of the protein, and there's kind of a normal version of that protein. And what happens is that abnormal version can convert the normal protein into the abnormal. And it causes all kinds of awful symptoms. And when they look at the brains of these people, they look somewhat like an Alzheimer's patient might. You have these plaques, or basically holes in your brain. And they're pretty nasty, and they're extremely resistant to heat, to chemicals, so they're very hard to kill. So it's not like with Salmonella or E. coli in your beef and chickens that you can cook it and get rid of it. You can't cook it and get rid of the infectious protein because they are very resistant to heat and other chemical measures that we think of as killing some organisms.
Now, let's go back to bacteria for a second. I want to talk about the players in this.
Okay.
Bacteria are mostly harmless. And not only harmless, most of them are helpful.
Yes.
So tell us about the good stuff, because we just said a bunch of bad stuff.
Right. Well, the good stuff is without bacteria, we wouldn't have some classes of antibiotics, like streptomycin. Actually, is named from Streptomyces. It's a genera. It's a soil organism, and these organisms excrete this antibiotic when they're in the soil as a part of their defense. And so they kill other bacteria, so they can have their own kind of little colony and niche where nothing else can invade them. And other good bacteria are the ones in your gut. They help you to digest certain things. They're also the ones that give you gas, but they do help with digestion. Cows, they rely heavily on their gut microflora because that's what helps them digest all of their stuff. See what else? Yeast, of course, if you like your occasional wine, but that's not a bacteria. But just to give you an idea of the benefits of all microorganisms, there are some that they're looking into for cleaning up oil spills and things like that. Mm-hmm. So there's lots and lots of uses, good uses for them.
So what about things like antibacterial hand cleaners, like soaps and household cleansers? Are they bad news then? Because we use them so that we can kill the bad bacteria.
Right. I think they have good and bad uses. You don't want to. The thing is, Staphylococcus. We hear about staph infections all the time. Well, that is just on your skin all the time, and most of the strains that are on your skin are fine. They're not going to cause a problem. You get a cut, you're not going to get it's not going to be the flesh-eating kind. The problem with overusing these, like with antibiotics, is that you will eventually have some strains that pop up resistant to this stuff and can become dangerous. And soap is a detergent. Detergents will kill bacteria. So really just a good hand washing is all you need, because it also washes off bacteria as well that are on the very surface. And those are usually the ones that are going to cause infection of some kind.
Okay, so would you say that we don't actually need to use the hand sanitizers and such? Just because I'm thinking during this entire H1N1 flu, debacle,
right.
We've been told repeatedly to use Purell.
I know, and my understanding, and I could be wrong on this, is that those are antimicrobial, not antiviral. If you look at the ingredients, it's ethanol, which is alcohol. Ethanol will kill bacteria, but it won't affect the virus. Now, what it can do is maybe loosen up the virus. It's maybe on your hands, so it washes off easier. But I'm pretty sure that those Purell and everything will not actually kill the virus.
So we're screwed. Good. Okay.
Pretty much. But washing your hands will remove the virus so that you're not spreading it. Because if you have a few virus particles on your hand, that's not really enough to infect somebody else. It takes, a viral load or a lot of viruses, unless it's an especially virulent strain of virus.
And when we will talk about those virulent ones soon.
Okay.
But we have a very interesting question submitted.
Okay.
Go ahead, Ryan.
Is there a lower limit on an organism's size? Why? Is there an upper limit on the size of a single-celled organism? Why? Why can't I have a square-foot pet amoeba?
Wow, those are really good questions.
I know, that's a really good question.
Well, even within the bacterial family, there are a range of sizes. I'm not sure what the lower limit of that would be. It probably has to do a lot with the cell membrane, what surrounds the cell and keeps all the cytoplasm and all the junk inside the cell. You have certain processes that have to go on across that cell. You have to be able to import, export nutrients like oxygen, salts, and things like that. So there may be a limit on how small that actually has to be in order to have those surface proteins on there to be able to do that exchange of nutrients and waste in and out of the cell.
Right.
Like viruses, they are very, very small, but they're host dependent. They can't live outside of the host. So that is one constraint. If you're too small, you don't have the cellular machinery to have your own metabolism. And as far as being square, you don't see a lot of square anything really living. It probably has more to do with the surface area and surface tensions and things like that.
Not
my best guess.
Why it couldn't be square. I think he wants to know why he can't have a large pet amoeba. Yeah.
Well, they might be messy because they're just kind of oozing all over the place. But, I don't know. I've never thought of that question before.
I think we should all do some research on why this individual can't have a square-foot pet amoeba. There's another question from the website.
What is the deadliest microorganism on Earth, and how scared should I be?
The deadliest. Well, I guess it might depend on where you are and what your health is. I think, to me, one of the scariest ones is Ebola. And I don't know why that one is so infectious. It's got an incredibly high mortality rate. You basically just bleed to death. You bleed from your nose, you bleed from your eyes, you bleed from every pore in your body, basically. I'm sorry.
No. That's why. Why?
I know.
How does that work?
What, the Ebola?
Yeah.
A lot of it is basically your own immune system trying to purge it. Everything becomes leaky. Your vessels and everything just kind of leaks because it's trying to get certain fluids and everything into and out of your cells. And so once your immune system takes over, sometimes it's your own immune system that can kill you and not necessarily the organism itself.
Right. Oh, that's highly disturbing.
So the hemorrhagic fevers are generally the ones that scare me. Fortunately for us, they're usually in Africa, places like that, for some reason. Seems to be a hotbed of emerging viruses.
Does that have anything to do with nutrition, or what would be the reason for that?
I think it's just a nice soupy place. That's the only best guess for me. And also you've got the apes and everything who are very similar to us. And if a virus emerges in them and then passes to humans, it can change enough to be quite deadly. And a lot of it is lack of hygiene, not clean water. And so once these things do emerge, they can be hard to get rid of because they don't have the hygiene. The good thing about it, if there is a good thing, is that they are so deadly and so virulent that they basically kill everybody and it can't spread anymore.
You are a cheery, cheery woman.
I know.
That's at least people die. So, audience, all your strangest microbiology questions answered right here by Lisa Hammett. Call in at 780-492-2577, extension 1. You can also email them to live@skepticallyspeaking.com or tweet us at Skeptical Radio. So let's continue talking about the immune system because I'm fascinated now. So does it react differently to things like pathogens than it would to viruses?
Yes, it will react differently. Bacteria come in and you have a set of cells that it will recognize that organism. And there are certain molecules in your body that will they flag that organism for destruction. And so you have antibodies that can do this. Macrophages, which are a special type of cell that will eat the bacteria and break it up and then take the broken up pieces and put them on the outside of the cell. And then your body will recognize those pieces and be able to basically seek and destroy other organisms. Viruses, because they are what's called intracellular, is they actually get into the cell. That's a little bit different and has kind of a whole nother set of cells that deal with viruses. So yes, your body does do different things depending on whether it's a bacteria or a virus.
Okay, and the way we know our immune system has kicked in are things like, the runny nose and the fever and such. So is it different for viruses than it is from bacteria, or?
It can depend on the virus. I mean, like when you get a cold, that's a virus. Mm-hmm. You feel awful. You have the runny nose, but you don't always have a high fever. Mm-hmm. Now, if you get the flu, which is also a virus, you may get all those other symptoms in addition to a fever. It depends on what chemicals get released in your body in response to that virus. And so there is a chemical called, I think it's interleukin-1. And it goes to your brain where your temperature center is, and it sets your thermostat higher. So that's when you get the fever. And you get the fever because your T cells and your immune cells are more active at that higher temperature. And it can also cause the infection some problems. So you've got your T cells that are working better and possibly causing raised heat causing problems for the whatever infection you've got. So a little fever is not a bad thing.
Okay, now how about to a prion or a prion? How does your immune system respond to that? Because it's a protein.
Right. I don't know that.
Would it not be your immune system that would respond?
I'm not up on all the research with prions. They're kind of hard to deal with. I don't know if you can actually. It's not like you can grow them because you can't really grow them. So how do you find the research for them? So I don't exactly know what the immune response, if any, because when you hear people getting mad cow disease, you don't hear of them getting sick.
Right.
They're fine, and all of a sudden they start having these muscular neurodegenerative problems.
So audience, any microbiologists out there that know about prions, call in. It's 780-492-2577, extension 1. You can email us at live@skepticallyspeaking.com or tweet us at Skeptical Radio. And I believe we have an email.
This email is from Kylie Sturgis. You mentioned Creutzfeldt-Jakob disease, and it's a prion disease, which apparently even the bad astronomer finds scary. Just how worried should we be about bacteria? And in addition, is it true that kids are getting too removed from getting into dirt, and the cleanliness of modern day is hurting their ability to build up immunity? This was in a recent episode of House, so it's getting into pop culture. And thanks, fantastic show.
Well, addressing the aspect of kids, I think so, a little bit. I think we're getting a little bit cautious about it. The way that our immune systems mature when we're children is being exposed to some of these. Now, I'm not saying that we should go out and stop vaccinating and so we all get measles and chicken pox and everything. That's kind of a different topic altogether. But a little dirt on your hands is fine. Your body learns how to recognize these microbes. Are they going to hurt you or are they not going to hurt you? And so getting out in the dirt and playing is fine. You want to come in and wash your hands and take sensible precautions. But I think that
coating everything in an antibacterial film, is
right.
bad.
And one thing I had read somewhere that one reason maybe allergies are so prevalent now is because we don't have parasites. We don't get parasites anymore, and there's a particular antibody called the IgE, which would fight these parasites that you would get. And IgE is also related to allergies. And so maybe now that this IgE isn't reacting with the parasites, it's kind of like trying to find something to do. And so then your immune system becomes overreactive in that way.
Right. Now, what was the first part of Kylie's question? It was how scared should we be about what?
About bacteria in regards to the prions.
It's back to the prions.
Back to the prions.
Because they're frightening.
Yeah, well, prions are completely separate from bacteria. They're just a protein. They're kind of their own class of thing, and they're not really associated with the bacteria. Like you can't get the prion from a bacterial infection. You can only get it from eating meat or infected meat that has the prion in it.
So vegetarians are safe?
Sure, they're safe from prions, but there's all kinds of other good things that can infect them too.
Okay. I was trying to help my vegetarian friends right there.
Yes, they would be safe from the prions.
All right. So we were talking about virulent viruses. Why are some worse than others? For example, the bubonic plague.
Well,
why was that so bad?
The bubonic plague, I believe, was actually caused by a bacteria called Yersinia pestis. But viruses, like, for instance, let's just take the H1N1 because they're talking about how virulent and infectious it is. Some viruses just are able to adapt to our immune system. They are able to find ways around it. And H1N1, for example, was a very novel virus. And nobody had any kind of protection against it. So once you get it, you get sick, and then everybody can get it because we don't have any protection against it. And then every time, for each passage or for each time it gets passed on to a person, the virus can learn, quotation marks, how to get around your immune system even better. And so they get very efficient at evading your protection, what your body's trying to do.
So why was H1N1, or why is, I should say, H1N1 so much more of a problem than the regular flu?
Because it is so new. Every year you go and you get your seasonal vaccine, and that's just kind of a variation on the same flu virus that you get every year, or goes around every year. And they just try to anticipate what changes will occur. The H1N1 just popped up all of a sudden, and it was novel. We hadn't seen it, they think, since around the mid-1950s, where a similar one went around. Mm-hmm. And so nobody's been exposed to this one before. It was so different, a strain, than everything else that we're used to seeing.
Well, let's continue to talk about vaccines for a minute. But every time I mention vaccines on this show, I definitely
get a flood of
calls.
emails, don't you?
I do. So I'm going to do it.
Okay.
The number is 780-492-2577, extension 1. You can also reach us at live@skepticallyspeaking.com or on Twitter @skepticalradio. So tell us how vaccines work.
Well, you have vaccines for bacteria and viruses, and they essentially work the same way. What they do is they kill or severely handicap the organism so that it doesn't cause, it's still alive, but it won't actually cause you to get the infection. So they take that, inject it into you, and it allows your body to respond to that. And when your body has a response to any kind of infection, it creates a memory response. Some of your cells become memory cells. And what they do is they circulate in your blood for the rest of your life, and if you get infected with that same agent again, there's an immediate and rapid response. In fact, it's so fast and so immediate that you really don't even know you've been infected again, because it purges it from your body before any kind of reaction occurs. And so that's what it does. So if, say, you get measles or chickenpox vaccine or the H1N1, it just exposes your body to that, to the parts that cause your body to respond, because there's only certain parts of the organism that your body recognizes. And so they expose your body to those parts, and it remembers it for the next time.
So I believe we have a call. It's Randy from Edmonton. Go ahead, Randy.
I just had a quick question about vaccines. If a person gets a virus, first of all, when the virus leaves their body and moves on to the next person, is it sometimes does it adapt every time, or does it get stronger?
It can. For each passage that it goes to, for each time that it goes to the next person. So if you get the virus and you pass it on to a relative, it may not change much, maybe a little bit, but you won't really see much change in the symptoms. But the more times it continues to go through the population, then yes, it can adapt and change and become more virulent.
All right, thanks, caller. So how does a vaccine trigger the appropriate immune response without actually infecting the patient with the disease in question? Because we hear this all the time.
They take the parts of it, like, for instance, a killed vaccine. They just take the organism and they kill it. And so there's only certain what they call surface antigens that are on the cell surface itself. And it could be a sugar or a protein or something like that your body recognizes. You got to remember, it's not necessarily the organism that's causing your symptoms. It's your own immune system fighting the organism that's causing the symptoms. So you inject those pieces into it, and your body will recognize those pieces. But since the organism can't actually multiply and replicate and get into your lungs and continue to cause the same immune processes to keep on going, you don't get sick from it. And then the other one, what's called the live attenuated or live killed. I'm sorry, not live killed, live attenuated. Mm-hmm. The organism has not been killed, but they have handicapped it to the point where it's not going to cause the disease. And I say not going to in almost all people. Some people may have a little fever or something, and some people may react to the vaccine. But for the most part, most people will never even know they've gotten the shot except for actually getting the prick.
Right. Well, so let's talk about those side effects for a second, because now obviously autism is not even remotely linked to vaccines. I'd like to start with that.
But in the scientific community, there is no controversy.
Thank you. So what are the actual realistic risks?
You can actually get the disease. For instance, there are very, very few number of people who get polio from the virus. But mostly, if you're going to react, it's going to be possibly an allergic reaction, because you can't actually be allergic to the virus or allergic to something in the vaccine. Some people may get a high fever, may become lethargic, and this is usually in children. We're talking about children, child vaccinations. High fever, like 104, 105 kind of high, just lethargic. And the high fever can cause neurological issues if it's not taken care of. And so I think that is part of what happens is parents go and they get these, you get your child vaccinated, and then they may get this really high fever. And if you wait around for too long, that can cause damage.
Right.
But for the most part, there's that, and that's just very, very few people. But if
when we're saying very, very few people, what kind of very few are we talking?
I'm not sure what the numbers are, but it may be like less than 10 per 100,000 or more.
Okay.
But again, if it's you or your child, it is a difficult situation.
Mm-hmm. So I'm leaving vaccines,
yes.
Which are pre-illness, and going to antibiotics, which are post-illness. So how does an antibiotic work?
An antibiotic only works on bacteria, not on viruses, not on prions, not on a fungus, but on bacteria. And there are several different classes. There's the penicillins, the cephalosporins, the aminoglycosides, and each one of them has a different action. Some of them interfere with how the bacteria makes its cell wall, the thing that keeps all the stuff inside the cell. Mm-hmm. Some of them interfere with how they make proteins. Some of them interfere with how their DNA replicates or how their RNA works. Some of them inhibit like mitosis or how they divide. So they have various ways, and each class of antibiotic will work differently. And some will work on certain classes of bacteria and some won't. That's why you have what's called like a broad spectrum. And those work on a lot of different bacteria, including the good bacteria in your gut. And so some people wonder, why am I getting sick? Why do I have diarrhea every time I take antibiotics? It's because it's messing with not just the organism making you sick, but it's messing with the good gut flora that helps keep you from not having diarrhea and everything.
So you mentioned before the antibiotic-resistant superbugs. Mm-hmm. So what does that mean?
That means that the class of antibiotics that previously worked on them won't work on them anymore. They have found a way to inhibit or prevent that antibiotic from working on it, whatever the action of that antibiotic was. They found a way to keep it from working.
So where do we go from there? Like, what can we do at this point?
At this point, scientists are scrambling. They're trying to figure out what to do.
Why does a bacteria become resistant in the first place?
Well, they can gain the resistance from another bacteria. Bacteria have two groups of DNA. They have what's called the chromosomal DNA, which encodes for all the stuff they need for day-to-day life, just metabolism, things like that. Then they have these very small pieces of DNA called plasmids. And it's these little pieces of DNA that carry the antibiotic resistance. And these little pieces of DNA can kind of come in and out of the cell, and they can go from, say, one bacteria that's not really causing you any problems, but it's resistant to this antibiotic, and it can kind of go in to the bacteria that you don't want to be resistant. And so now this other bacteria that previously was not resistant is now resistant because this little piece of DNA came into it. So that's one way. The other way is just mutation. You got to remember there's billions and billions of these little things constantly replicating and changing and mutating. And so just small mutations along the way can create this antibiotic resistance for that organism.
Again, the number here is 780-492-2577, extension 1. You can also reach us at live@skepticallyspeaking.com or on Twitter @skepticalradio. And we'll be right back with those questions and more on microbiology right after this.
Next week on Skeptically Speaking, we'll be discussing the paradox of choice with psychologist and author Barry Schwartz. Dr. Schwartz contends that although it may seem like you want more choice and more options and more variety, and that having those myriad alternatives will make you happier and more satisfied, you're wrong. It's actually making you miserable. Find out why on the next Skeptically Speaking, every Friday at 6 p.m. Mountain Time here on CJSR.
Welcome back to Skeptically Speaking. I'm your host, Desiree Schell. Before we get back to Lisa Hammett and microbiology, I have two announcements. First, the next 2 weeks' shows will be pre-recorded as the team is taking a well-deserved break over the holidays. Those of you listening by podcast or on one of the lovely stations that rebroadcasts us probably won't notice a difference at all. But to those wonderful people who listen weekly on Ustream, you have the next 2 weeks off. And Professor Richard Wiseman, author of Quirkology and the Luck Factor, will be giving a lunchtime talk while he's in Toronto promoting his new book, 59 Seconds. If you haven't heard of Wiseman, Google him and you will be duly intrigued. So on Monday, January 4th, from noon to 1:30, head to the Fox and Fiddle at 27 Wellesley Street East. The talk is presented by the Association for Science and Reason. And for more details, you can visit the events section at scienceandreason.ca. So, Lisa, we hardly have any time left, but I want to talk about what's new in microbiology.
Which part? Immunology? Virology? Or what?
You're killing me. What's most interesting? Let's go with that.
Wow, there's so much. To me, some of the most interesting things going on have to do with the stem cell research, and they're finding ways around using embryos for stem cells. And in fact, that was one of the top 5, I think, cited research papers this past year was about finding ways to not use embryos to get stem cells.
Right.
And they've actually been able to start doing that. I know there was some controversy. I think it was a Chinese, wasn't Chinese, a scientist that he actually fabricated data about being able to take a cell, a somatic cell, or not an embryo cell, and make it so that it could be turned pluripotent or go into any kind of other cell. So to me, that's the most exciting thing, one of the most exciting. There's just so much. It's hard to pin down really anything, one particular thing. I think some of the more exciting stuff, in my opinion, is I love reading about evolution, is some of the showing at a molecular level how things have evolved. For instance, the immune system or anything really.
And there's been recent studies on that?
There's studies all about this all the time, yeah, about how this stuff has actually evolved, how particular systems, like the immune system, how did it all start? So yeah, there's a lot. There's just so much.
Well,
it's just amazing.
I have one that was submitted.
Okay.
Actually, I'm sorry. I'm taking Ryan's job right now. Ryan, do go ahead.
Okay. We've discovered water ice on Mars much closer to its equator than we thought. We've also discovered that something on the planet is producing methane. Should we begin getting excited yet?
You see why I wanted to read that myself.
Yes, it's very interesting. And actually, I heard about that a while back, a couple of weeks ago. I thought, wow, that is really cool. That is exciting, possibly. What they don't know is if it's biological or if it's geological. Methane is what you call an organic molecule. An organic molecule just has a carbon with some hydrogens attached to it. And so methane is CH4, sorry. So it's a carbon with some hydrogens. And we have bacteria, organisms on this planet that produce methane as part of their metabolism. And so the thinking is that maybe some of the conditions underground on Mars are similar to some of the conditions on Earth, billions of years ago. And so the hope is that there may be some organisms under there that don't require oxygen that are producing this methane. So they're going to have to look at there's different kinds of hydrogen. There's heavy hydrogen and light hydrogen. Biological systems on Earth tend to like the lighter hydrogen. So if they look at this methane and say, okay, this methane coming out has lighter hydrogen than, say, water or other hydrogen on Mars, then it might be biological. Or it could be geological. But either way, it's exciting because it's either geologically active or biologically active, which they didn't think was either one of those was true on Mars. So,
yeah, I would love to talk about this for longer, but that's our time. Thanks so much for being here, Lisa.
That was fun. Thank you so much.
So you can visit our website for the comments section of this episode and for links to our very joinable Facebook page and our extremely followable Twitter feed. There's also an iTunes link so you can conveniently subscribe to the show. So thanks to everyone who sent in questions, and thanks to the rest of you for listening. And we'll see you next week in a pre-recorded way on Skeptically Speaking.
South America does not exist. The moon is painted on the sky. Things sometimes fall up, but you never see. And everything you believe is a lie.
Dinosaurs were invented by God as a test. Our bodies don't rot when we die. Oh, the hippies are working for the government, and everything you believe is a lie. She